Welcome to this AnaBios webinar on advancing analgesic drug discovery with a novel translational strategy. I'm Andre Ghetti, the CEO of AnaBios Corporation. This presentation will be about 25 minutes long, and we will have a Q&A session at the end during which you will have the opportunity to ask questions through the chat box. Traditionally, non-clinical drug discovery has been conducted using animal models under the assumption that the results obtained in these models are translatable to clinical findings. Unfortunately, this is very often not the case, as proven by the high failure rate of developmental studies in clinical settings. Specifically, in the case of pain drug discovery, the situation is further compounded by the patient population heterogeneity. In this study, for example, different groups of pain patients affected by polyneuropathy, radiculopathy, peripheral nerve injury, or post-herpetic neuralgia were profiled using quantitative sensory testing. And as you can see, there are clear differences across the different patient populations, but also within each group of patients, within each specific etiology, there are differences in sensory loss, thermal hyperalgesia, and mechanical hyperalgesia. So the question is, how do the preclinical animal models of pain map onto these very diverse patient populations? And unfortunately, we don't really have a good answer, which means that in most cases, the results obtained in animal models cannot help us predict which specific therapeutic indication could be most appropriate for a given drug. AnaBios has decided to tackle these translational challenges by introducing a new paradigm, which enables the study of drug potential efficacy and risk directly into human cells and human tissues at the preclinical stage. Now, there are some fundamental challenges in trying to utilize human cells and tissues for investigational studies. The first one is ensuring sufficient viability and quality of the study material. The second is access that is consistent and high enough to ensure proper support of drug discovery program. And the third one is reproducibility. So with regards to viability, we have spent a significant effort in the early days of our R&D work to develop proprietary solutions that prevent cold ischemia and reperfusion injury. And this allows us to procure organs, tissues, and cells from a variety of locations within the United States, essentially canceling the effects of shipping time. Access is ensured by a very extensive network that we have established with partnering hospitals and transplant centers within the United States, which allows us to receive many samples on a weekly basis for a variety of organs and tissues. And reproducibility has been accomplished by standardizing the recovery methods across all the centers that work with AnaBios. Also establishing very rigorous quality control procedures, and also having access to medical social history for the organ and tissue donors, which allows us to essentially match the different tissue types, in particular the ones from healthy donors versus diseased donors. There are in the flow of this type of operation, there are several steps that are critical to the overall success of the paradigm. So we start with the organ donors for which we have received ethical consent for recovery of organs and tissues for research. And we procure a variety of organs and tissues, hearts, lungs, sensory ganglia, liver, and spinal cord. And all of these samples are extensively handled at the hospitals in such a way that we can perfuse our proprietary solutions which stabilize the samples during shipping. And then using the same logistics that are utilized for transferring organs and tissues for transplant, we ship all of these samples to our laboratory in San Diego. Once we receive the organs and tissues in San Diego, we can start performing either tissue or cell-based assays for assessing in a functional context the potential effects of a drug on the tissue of interest. Given the viable state and high quality of the samples, these are also excellent study materials for conducting genomics or proteomics type of investigations or other biochemical type of studies. There are several advantages in utilizing this approach. The first one is that we have seen through the work we have done with our clients that this is really predictive of clinical outcomes. And there is a significant effect in lowering the developmental risk related to interspecies differences. The other aspect which is extremely powerful in this type of approach is the ability to study drug action in the context of pathological states. And this is really something very important because using animal models, one has to create hypotheses and generate an artificial model of a human disease, which often is inaccurate given the limited knowledge we have of many of these diseases. So in the case of human tissue, for example, sensory ganglions from donors affected by chronic pain, we don't need to make any assumptions or hypotheses as to the specific mechanism of the disease. We simply utilize the tissue obtained from the diseased donors. Another aspect which is very important and has very significant practical implication is the fact that this paradigm can be quantitatively predictive. That means that the potency measured in these human ex vivo cellular or tissue preparation is truly predictive of the activity of the compound in the human body. And so these data can be extremely useful in designing the first-in-human studies and in particular in defining the dosing. In the context of pain drug discovery, we focus on the peripheral nervous system by procuring the dorsal root ganglia, and we have access to the ganglia from the T1 level all the way to S1. We procure ganglia bilaterally, and we utilize the ganglia for establishing mixed glia neuronal co-culture. So we dissociate the cells in the ganglia and establish co-cultures which can then be used for electrophysiology-based or imaging-based type of studies. In the early days, we've spent a significant amount of effort characterizing and validating these cultures. These are data that we generated in collaboration with the laboratory of Rob Giraud at Washington University. And we were able to show that the phenotype of the cells in these cultures is stable for about 2 weeks. And the cells respond to algogenic and pruritogenic agents as would be expected for human sensory neurons. Over the years, we have used these preparations to study the effects of compounds that were targeting a variety of ion channels from voltage-gated to ligand-gated channels, TRP receptors, glutamate receptors, GABA receptors, opioid receptors, and others. And essentially, any methodology that it relies on electrophysiology, whether it's current or voltage clamp, or the standard imaging methodologies based on fluorescent indicators can be utilized in the context of these preparations. The cells can also be transfected or infected with viruses, so it is possible also to express exogenous genes of interest in these cells. Now, one thing I want to bring your attention to is on the left side of this slide. There are 3 action potentials recorded from 3 different neurons. And as you can see, the kinetics of the action potentials are quite different. And this points to the heterogeneity of cells in these preparations. And so it became clear very early on that it was necessary to add methodologies for investigating drug effects that could sample fairly large cell populations in a short period of time. And this is difficult to do using electrophysiology-based methods where only one cell at a time can be interrogated. And so we developed some methods to study human sensory neurons in cultures using imaging. And this one in particular relies on the employment of field electrical stimulations to activate voltage-gated channels. We were especially interested in characterizing and profiling voltage-gated sodium channels, which in these days are an important target for analgesic drug discovery. So in this specific assay, we use electrical field stimulation protocols that have been calibrated in such a way that we can selectively stimulate voltage-gated sodium channels without reaching the threshold for direct activation of calcium channels. And so what happens is that our field stimulations opens the voltage-gated sodium channels. This leads to cell depolarization, which in turn leads to a secondary activation of calcium channels, and the calcium influx can be detected using a fluorescent calcium indicator. So we have an assay that can rely on calcium signals for actually tracking the activity of voltage-gated sodium channels. As you can see in the bottom left part of this slide, two cells represented with the red and the blue trace actually have different patterns of responses to a train of electrical field stimulations. And as it turns out, the two cells actually are expressing different subset of voltage-gated sodium channels. The responses to electrical field stimulation delivered following these protocols also can show differences from cell to cells with respect to sensitivity to TTX, which is a toxin that blocks only a subset of sodium channel subtypes. But all responses are blocked by lidocaine at a concentration of 200 micromolar, clearly indicating that all the responses are due to the primary activation of voltage-gated sodium channels. Now, using combinations of TTX, activation threshold, as well as the response to different frequency of stimulations, we have been able to distinguish 6 classes of cells based on specific combination of sodium channel subtypes expressed in each one of the cells. And what is interesting in these methods is that we can then stop the electrical field stimulation and follow on with application of a variety of agents, for example, capsaicin, mustard oil, and cold buffer. And as shown in the bottom right portion of the slide, we can then characterize cells for their nociceptive profile. So there are cells that respond exclusively to capsaicin, other cells that respond to all 3 agents and are sort of multimodal nociceptors. And so these data can generate a very complete and complex phenotypic profile for all the cells in the field of view. And so we can generate these phenotypic profiles for 50 or 100 neurons in a single experiment. And what is shown here in the pie chart at the center of the slide is that we can identify the different classes of nociceptor in each preparation. And for each nociceptor in the bar graphs, we have the specific profiles of the sodium channel related responses that have been measured using electrical field stimulation. And as you can see, the different nociceptor subtypes express different arrays of voltage-gated sodium channels in such a way that they have distinct profiles of responses to the electrical field stimulations. And what is really interesting is to then superimpose to this type of picture the effect of a particular drug of interest, so that one can determine not only if a new drug candidate is active in human sensory neurons, but if there is a specific population or subpopulation of sensory neurons that is sensitive to the new drugs. And again, this is important information that can help understand what may be the most appropriate therapeutic indication for a particular drug. The other important aspect is that the data that I'm showing in this slide is collected from cells from a healthy donor, non-pain donors, cultured under normal conditions. And this picture changes quite significantly in pathological states. Now, I mentioned that the investigation of drug effect in the context of pathological states is very important for obvious reasons. And we have a couple of ways to do this using the human neuronal preparations. The first one is to take cells from donors that were not affected by pain states, culture them first under normal conditions, and then apply a challenge with either an inflammatory agent or an agent known to induce neuropathy, like a chemotherapy agent. And so what you see in the top row of the slide is that when we treat, for example, cells with prostaglandin E2, we increase the excitability of the cell itself. And so the same stimulus that was previously generating a single action potential can now generate a train of 3 action potentials. And similarly, if we treat the cells for 24 hours with oxaliplatin, what we see is that in response to a cold gradient, both the number of cells as well as the thresholds at which the cells start responding to the application of cold buffer shifts in such a way that there is an increased responsiveness which may correspond to the cold allodynia and cold hyperalgesia, which is often reported by patients treated with oxaliplatin or other chemotherapy agents. So these are what we call the in vitro pain models, and these are readily obtained starting from normal DRG cultures. The second possibility to study pathological states is to use DRG neurons isolated from donors that were known to be affected by one form or another of chronic pain. And as I'm showing in the bar graph at the bottom left, about 20% of the DRG cases that we receive are from donors affected by pain. In the bottom right, I'm showing a recording of spontaneous activity, spontaneous action potentials measured in a neuron from a chronic pain donor. And this is something that we see especially for particular donor types, and is completely absent in recordings from normal DRG neurons. So both of these strategies are extremely useful for understanding the potential of a novel drug candidate in treating specific pain states. First, for distinguishing the activity of the drug in the context of inflammation versus neuropathic states, and using DRG from pain donors, really understanding for specific disease states if the drug can have the ability to reduce neuronal excitability. So to provide a few practical examples of how this strategy can be used in evaluating compounds, I'm showing in this slide recordings of action potentials from an individual neurons. And as you can see from the top portion of the slides, we can stimulate neurons using trains of action potentials. In this case, we're using a train of 60 action potentials generated by delivering stimulations at varying frequency from 1 Hertz to 10 Hertz. Now, we have established empirically that in this assay, when a compound results in inhibition of at least 30% of the action potentials, this corresponds to a level of activity which translates into analgesic effects in patients. So in this particular example, a proprietary drug, which we'll call drug A, tested at its IC50, results in significant inhibition of action potentials, especially at the higher frequencies. This particular drug is a blocker of voltage-gated sodium channels, and therefore the frequency dependence is not surprising, given that most small molecule sodium channel blockers are state dependent. What is especially interesting in this series of experiments is that when you repeat the test on cells that have been sensitized using a mixture of prostaglandin PGE2 and bradykinin, we see that the drug actually has a more potent inhibitory activity. And the data are summarized in the bar graphs on the right side. And if you look at the blue bars, which corresponds to the effect of drug A, you can clearly see that moving from the control state with the normal cultured neurons to the inflammation state, there is a significantly more pronounced inhibition of the action potentials. The two other drugs, drug B and C, did not show a similar effect. Now, looking at the same type of measurement run with compounds that are known and have well-defined clinical outcomes, we can really see the value of this type of approach. So what I'm showing here is essentially the same type of testing run using roxatrigine or carbamazepine. These are both sodium channel blockers that have been shown to be effective in treating trigeminal neuralgia. And so we're reporting the percentage of inhibition of action potentials, a percentage actually of remaining action potentials in a long train induced artificially through current injections. Again, we're testing the effect of the drug at different firing frequencies. And as you can see for both drugs, at concentrations which correspond to the free plasma therapeutic concentration, we see that there is enough inhibition at the highest frequency to predict analgesic efficacy, which indeed these compounds have shown in clinical settings for trigeminal neuralgia. Now, what is interesting is to look, for example, at roxatrigine in a different pathological context. So if instead of testing on normal DRG neuronal cultures, we repeat the same test on cells that have been sensitized with prostaglandin PGE2 and bradykinin, the result is quite different. And as you can see, roxatrigine now is much less effective in inhibiting action potentials. And at the free effective therapeutic plasma concentration, there is barely any reduction in the action potential firing. And this is likely due to the fact that this compound is not particularly effective in inhibiting the sodium channel subtype NaV1.8, which is known to be upregulated and be critical in action potential firing under inflammatory conditions. Similarly, a compound that was originally developed by Pfizer, PF050897771, which is also a NaV1.7 selective compounds, devoid of NaV1.8 activity, shows very little inhibitory action under inflammatory states. Now, both molecules were indeed tested in clinical studies in pain conditions that are associated with significant amount of inflammation. For roxatrigine was a low back pain patient population, and for 771, it was tested in patients that had undergone third molar extraction. And in this clinical trial, both molecules failed to show significant analgesic effect. Again, not surprising in light of these data and in light of the fact that they do not have any NaV1.8 blocking activity. By contrast, when we look at, I will show later, the profile of a compound with NaV1.8 selectivity. We also did test roxatrigine directly in DRG neurons from donors that were affected by low back pain, again, confirming the results of the in vitro pain inflammation model. We see that roxatrigine in low back pain DRGs shows very little inhibitory activity. Now, for a NaV1.8 active molecule. We looked at a molecule that was originally discovered by AbbVie, A803467. And as you can see, contrary to the prior examples, this molecule has more inhibitory activity under conditions of inflammation. We have conducted a significant number of validation studies using these preparations, and I encourage you to visit our website where you can download all of these peer-reviewed articles or posters that we have generated over the last several years. A question that often comes up is at which stage can be most effective to utilize this translational paradigm. And we have seen companies benefiting from this strategy at multiple stages in the discovery pipeline, from early target identification, where especially relying on tissue sample, DRG samples from healthy or pain disease donors can really be extremely helpful in identifying or validating novel potential targets. And then both lead identification and probably in particular lead optimization and clinical candidate selection is probably the sweet spot for utilizing these methodologies and making sure that only molecules that have true potential in human are advanced in the pipeline. We have used also these approaches over the years to help companies that had compounds at clinical stages and in cases in which the clinical studies had to be halted due, for example, to unforeseen toxicity signals. And in some cases, we've actually discovered that they were actually overdosing the patients due to the fact that the dosing was all set based on the efficacy observed in rodents, for example, when humans were a lot more sensitive to a specific drug. Or in other cases, we've actually identified whether a toxicity signal was related to on or off target type of effects. Speaking of therapeutic window and off-target effects, I want to briefly mention that we have availability for conducting studies also in other tissues that are highly relevant for establishing the therapeutic window, especially when the target is an ion channel. So we can do studies both in isolated human primary cardiomyocytes or cardioventricular trabecular, and these investigations are extremely useful in understanding and defining the therapeutic window of a compound. So to summarize, I have shown you that with this new paradigm, it is now possible to study drug effects directly in ex vivo human models. This strategy enabled the study of drug action in the context of pathological states, which is important for truly understanding the therapeutic potential of the compound, but also defining in a high level of detail the specific therapeutic indication that should be used for the first proof of concept clinical studies. And the last aspect, which is also critical and has very practical implication, is the ability to predict the potency in human, thereby informing the selection of the most appropriate dose for the first in-human studies. And I will stop here and open the Q&A session. Thank you very much for your attention. Okay, so if you want to start sending in questions through the chat box.
Co-localization, whether he can just
sorry, Andy, your audio is going in and out. If it may be better to actually type your questions down below. So I have a first question that came in that is inquiring about the observed donor-to-donor variability in pharmacological responses. And so I will say that is relatively small. It depends in part on the specific pathway that one is investigating. Some pathways show a little more donor-to-donor variability, like opioid-related pathways, for example, or itch-related pathways. But overall, the responses are fairly consistent. Again, the biggest difference is observed between healthy, normal, non-pain donors and pain donors. So that's the first important distinction to make. In general, the variability is more related to the magnitude of a response or sensitivity to a drug. So we've never seen a situation in which a particular drug is an antagonist in one donor and an agonist in another. Yeah, so, and there's another question that still is inquiring about the variability on donor-to-donor variability, which I just discussed. But in addition, the timing culture that the DRG neurons are kept. So I mentioned earlier that we have run early validations on these preparations and have concluded that there was good phenotypic stability over the first 2 weeks in culture. The cells survive well beyond the 2 weeks time, but after 2 weeks we start seeing phenotypic drifts and changes in their physiological properties. So typically we conduct all the experiments and testing within the first 12 to 14 days in culture. Another question is, are these experiments only conducted in acute conditions, or can drugs or stimulations applied in long-term setting? It is possible to do relatively longer-term exposure. Again, we have the limitations of the 2 weeks time window, but it is possible, for example, to leave drugs on for extended period of times. We've done studies with companies that were interested in observing changes related, for example, to epigenetic or other expression-related changes in the cells. So another question is with regards to the specific preparation. Do we use screening platforms that are cell-based or tissue-based? These are all cell-based. So we're currently trying to develop additional methodologies that would utilize sort of the intact ganglia preparation. We obtained the ganglia with still attached a good length of the afferent fibers. So we're developing assay that would look at conduction of electrical impulses in those preparations. But for now, we're using exclusively isolated cells, and everything that I described today was relative to isolated cell cultures. There is an interesting question on any correlation between the magnitude of response in vitro studies and the magnitude of clinical pain grade. So we do have access to a good amount of social medical history on the donors. However, we don't have typically access to such detailed information as the grade of pain. So we know, for example, in the case of chronic pain donors, what type of pain had been diagnosed and the part of the body that was affected by the pain. So another thing that I did not mention, but we do, is once we identify the specific region affected by pain and presumably the dermatomes that were interested by the pathological states, we culture cells selectively from that area. Unfortunately, we don't have quantitative sensory testing or other reports that indicate specifically the pain grade and the pain intensity. And in correlating drug responses in vitro with clinical outcomes, what we've been able to do is essentially what I've shown in the presentation, where we have been able to tell with pretty good correlation which drugs seem to have a potential analgesic efficacy and drugs that don't. And so far, correlation has held very well with the clinical outcomes. Yeah, so there's a question on how many cells per ganglion we obtained. In part, it depends on the level of the ganglia. So we obtain from the T1 to S1, ganglia size as well as cell size changes quite a bit. But there are probably a few thousand cells that can be isolated from these tissues. The specific yields is also a function of the donor age. And clearly with the younger donor, it's easier to obtain higher number of cells. Older donors tend to yield a lower number of neurons. There's a question on whether or not only small molecules can be tested in these preparations or also biologics. And yes, absolutely, we can test biologics or small molecules. These methodologies are completely agnostic as to the nature of the drug. And there's one more question on the availability of DRGs on a weekly basis to support medicinal chemistry efforts. Yes, absolutely. So we receive sets of DRG, human DRGs, multiple times a week. And by virtue of the frequent procurement and the half-life of these preparations, we absolutely can support programs that require to test compounds on a weekly basis. There's a question on, can we detect the neuronal activity within 1 week after dissociation? The answer is yes. So with the caveat that these cells are not spontaneously active, at least when they are isolated from healthy non-pain donors and are not treated with special sensitizers. But we pretty much from second day in culture all the way to day 14, when we typically try to wrap up our work, we see the ability to respond to a variety of agents, have action potential in response to injection of currents and so forth. So 1 week is definitely within the time window. Okay, I think there is, at this point, no more questions. Again, if you have additional questions or want to learn more about this paradigm, please contact us, visit our website, and we'll be happy to give you additional information and details on the capabilities of these methods. Thank you very much for your attention.
Good.
Very good.
Yeah, you had quite a few questions.
Yeah,
you had actually
yeah.
more questions than